We're here today with Dr. Alan Ho. My name is Dr. Dave Seftel. I'm the Medical Director and Research Coordinator for the Macular Degeneration Foundation. What we'd like to talk about today is the overview of cell-based therapies for AMD and retinitis pigmentosa. You presented an excellent review of the area, and what we wanted to do is to get a sense of where these trials are positioned, what is the current status of those trials, and which are the most promising.
Okay, well, it's good to be here. I think currently there are several groups around the country looking at cell-based therapies for the treatment of retinal degenerations, and I'll speak more specifically in detail for advanced age-related macular degeneration. We've made a lot of progress on the wet form of the disease with injection therapies and a specific pharmacologic target. We know that the dry form, for example, geographic atrophy, is perhaps a little bit more complicated process of withering of the vision cells, the photoreceptors, and the underlying tissues like the RPE and choroid that support the vision cells. And cell-based therapies are one strategy to try and attack that problem. Two groups at our institution, at Wills Eye Institute, are using cell therapies to try and treat age-related macular degeneration and geographic atrophy. There's one group that's using embryonic cells placed under the retina in a traditional vitrectomy transretinal surgical procedure. The cells are introduced under the retina with a cannula, and a bleb of fluid is created, and the cells are placed there that way. We're using a different approach to try and reduce cells leaking from the hole. And this is that first approach was by the ACT group. We're with the Johnson & Johnson company that people are familiar with, using an adult umbilical tissue derived from the umbilical cord tissue cells placed under the retina through a microcatheter from the outside in. The current status is that both of these studies are preliminary, and there's no evidence that any cell-based therapy is a proven, safe, and effective treatment for atrophic AMD or for any other degeneration like Stargardt's or retinitis pigmentosa. However, we do have some preliminary data that I reported on here today on this meeting about the Janssen study showing some patients having some improvement, surprising improvement, that is endured, to our surprise, over 6 months to even longer, up to 1 year. We've also had patients that have had problems as we developed the surgical technique from retinal tear detachment. So it's not something that's ready for prime time, if you will, but we think we're getting better and safer in delivering the cells with a microcatheter into the subretinal space. People always ask about what's the mechanism for these cells potentially having an improvement in vision. And cell therapies are different than our concepts of drug therapies, where there might be a specific molecule and mechanism of action. In cell therapies, we think that the mechanisms are related to release of trophic factors or cytokines, little growth factors that are in the area of the diseased tissue, or by cell-to-cell interactions that might be supporting the cells that are degenerating. That's one hypothesis, that is, there is support of existing degenerating tissue. The other approach, of course, is perhaps what's people think of as in terms of stem cells, and that is taking a stem cell that can become any kind of cell, putting it in the area of diseased tissue, and then replacing those diseased tissues with functional cells of the same like. And in the one embryonic ACT trial, that might be the approach. In the Janssen trial, it's more of the trophic approach, or supporting of the degenerating tissues. So I think it's exciting. We have some preliminary data. We have certainly a lot more work to do to make the procedure safer and to scale up the trial, and we're continuing in the phase 1 study per the recommendations of our safety committee to look at this even further. But there there is some hope, I think. Patients with the dry form of AMD and retinal degenerations really don't have any current treatments, but they can rest assured that we're not standing pat. We're working carefully, scientifically, to try and make their lot better.
Let's talk about embryonic and then talk about adult stem cells. One of the issues in embryonic has always been rejection. And certainly in the spinal cord trials, they would get anti-rejection medication for at least 6 weeks. As we know, the spinal cord trials have been discontinued for a variety of reasons. When you use embryonic, are you using anti-rejection medication again for 6 weeks or for a much longer course?
In the embryonic cell trial, the ACT trial, I believe there are some medicines used to that effect. Now, let me just say this. The eye is a privileged space. We're very lucky to be working on a valuable site, valuable sense that people all recognize as important. And the eye is privileged from an immune standpoint. I'm more familiar with our Janssen study where we have the adult umbilical tissue-derived cells, and we don't put the patients on any immune suppression regimen, and there's been no sense of cell rejection, significant inflammation that would imply the body's kind of reacting to the cells. In fact, out of our initial lot of 25 patients, we have not seen any kind of immune response or tumor formation from the cells.
In other embryonic trials, specifically the spinal cord trials, the FDA had concern about neuromas, that is overgrowth of nerve tissue. Have you seen that in any of your patients? We have
not, as I said, seen any evidence of teratoma formation, neuroma formation. We see some areas of where, often we see nothing of where after the cells are placed, there's a bleb of fluid the next day or week that kind of goes away, the fluid goes away, we think the cells kind of disperse locally. We see some areas where we see a little bit of a thin band of fibrous tissue that may represent some maturity of the cells. We don't know if those cells are still active if we see some fibrous tissue, but we don't see any specific neuromas or other kind of tumor formation, if you will.
So switching gears to adult stem cells, one of the questions we frequently have is, are these adult stem cells actually rewiring? Because ultimately, if you have new cells wiring up to an existing nervous system, are they making new connections at a neuronal level? And if not, is it purely a neurotropic effect?
We don't have any evidence now of rewiring or differentiation of the cells that we place, these adult umbilical cells. We don't have any evidence that they establish any wiring or neurologic connections in either animal models nor in the humans. We think that the effects can be potentially achieved just by trophic effects or cell-to-cell interactions.
Are there any preconditions that you found in the laboratory that make it easier for stem cells to engraft and to proliferate appropriately? In other words, the medium that you're using.
Yeah, again, a lot of this basic science work, I would defer to the basic scientists. I'm a surgeon. I'm a translational clinical trialist, as I would view myself right now. And so, yes, there are conditions under which, for example, if you have a layer of diseased tissue, like the retinal pigment epithelium, you need a certain kind of membrane, like Brooks membrane, upon which cells that were meant to differentiate, for example, perhaps embryonic stem cells, there has to be a specific plane upon which these cells can spread themselves out.
You're now obviously working on surgical approaches, or to improve the surgical approaches to implant these cells as close to the critical area as possible. And I know that there have been other approaches where they try to inject the cells more generally in the area and use trophic factors to make them go to the site of action. Those haven't been particularly successful. Is there any work that you're doing in that area to make it less invasive? implantation?
That's a good point. Right now, we are placing the cells in the subretinal space, which is a challenging space to get into, but we think we're making some progress there. We use a microcatheter. A simpler way, perhaps, would be to just inject the cells into the vitreous. That might make for a safer procedure, but we are concerned that the cells, at least in the Janssen, Johnson & Johnson trial, might create membranes and scar tissue on the retina and create problems in that regard. Of course, there are other clever tacks, if you will, of getting cells into the vitreous cavity, like Neurotech has created this encapsulated surgical implant that houses cells then that can release specific growth factors into the vitreous cavity and therefore towards the retina. Those are some of the approaches. Currently, we think the subretinal space is where we need to be.
Right. And in terms of encouraging these cells to survive and to continue to function, there has been some speculation about the use of adjunctive electrical stimulation, external electrical stimulation. Is that something you're looking at this point, or is that a separate arm of study?
I think that's something that could come down the line if our preliminary results achieve some efficacy and safety for whatever amount of time. Clearly, we're talking about conditions that have or will be lifelong, advanced AMD, retinal degeneration. So, yes, we need to figure out ways to make the intervention more durable. I think what was surprising in our Janssen presentation was that the patients that did have some improvement, sustained improvement over 6 to 12 months. Again, small numbers, but the magnitude and the durability were surprising to us, and perhaps we might need less of the regeneration or support over time than we might imagine.
Now, in terms of measuring the outcomes, you obviously measure visual acuity, do you measure color perception? What are the things that you're measuring, the elements that you're measuring?
There are a lot of things we measure, and sometimes they're challenging in patients with significant vision loss, but certainly visual acuity, contrast sensitivity. You always want to get a sense of how the patients feel that they're doing with a visual function questionnaire. From an imaging standpoint, I think we have a lot of work to do. We certainly do photography, autofluorescence to measure areas of atrophy, angiography, OCT I mentioned, but we need to be able to, if we're seeing a treatment effect, begin to understand what's happening from a functional standpoint. We have some potential imaging technologies or testing technologies that might help us do that. But again, they're challenging in patients who, for example, can't fixate, multifocal ERG testing, etc.
Right, that was my next question. What is the role of multifocal ERG tests like that?
We need to figure out whether or not we can do those tests reliably in these patients.